Hello students, welcome to EPG Pathshala. My name is Dr. Ashish Pal. I am Scientist T at Institute of Nanoscience and Technology, Mohali, Punjab. I am going to discuss a module, self-assembly in nature, micelles, liposomes and capsules under the paper Nanobiotechnology. So the objective of this module is to know about different self-assembled structure like micelles, vesicles, capsules formation, and what are the underlying principle which actually governs this self-assembled nanostructure formations. So we'll look into more into details on them, and next is how to characterize these different nanostructure. That's very important for us to know, and we will learn different techniques as we go along in this in this module. And the third thing is that we would look into the size, shape, dynamics of all these nanostructures, and as we do it, we would also ask ourselves questions: which technique will be best for looking a particular nanostructure, whether it's a whether the technique needs to be non-invasive or invasive, if you have to, or if which technique is good if you have to look the entire overall structure, or you want to look core of nanostructure, or you want to look at the corona. So we would get to know as we go along, and this is going to be the main objective of this module. So we look at the self-assembly of amphiphilic molecule. What are amphiphilic molecule? Amphiphilic molecule means here you have both non-polar part and a polar part. Now polar part can be ionic, as you see in this case you may have sodium rhodesyl sulfate, you have C12 non-polar part, and along with that there is a head group which we call, there is a sulfate group which is a head group. It can be also CTAB based surfactant where you have C16 chain, but in the head group you have ammonium, trimethyl ammonium group present, or you can have the head group as pyridinium group, or there are some example like that, or you can have the head group as non-ionic group, something what you see in Triton X 100. You see here you have a non-polar group which is branch chain, but other than that you have head group is polar but non-ionic. So this can be represented in cartoon, this single chain with a head group or double chain with a head group. This is the hydrophilic group and the chains are hydrophobic. Or there can be also Bola amphiphile chain, that means both the side you will have head and in between the heads are connected by this tail. Or you can have a Gemini surfactant, so where your head groups are actually connected by a spacer. Now if you take this amphiphilic molecule which has a hydrophilic head group and hydrophobic tail, then and if you try to self assemble them, if you mix them, they can form different assembly pattern. It can form a monolayer on air water interface, where in water the hydrophilic head group will interact and the hydrophobic part will be flanking in the air. Or it can form a spherical micelles where all the hydrophilic groups are exposed to water and all the hydrophilic tail is inside. Or it can have planar bilayer, so it's the bilayer structure where the hydrophobic chains will interact with each other, and this hydrophilic head group, they will interact with the water molecules. Or it can also form these worm-like micelles. So spherical micelles, it is spherical, it can also become rod-like, worm-like micelles, or it can form, or the planar bilayer that might also get folded into a vesicle-like structure, or it can form inverted. So there are different aggregate morphologies. However, these aggregate morphologies are determined by the two opposing forces, which one is attractive forces, which are the hydrophobic effect, all the hydrophobic tail they want to come together, and repulsive force, that is the head group, if their head group is charged, so they would have a repulsion. So based on that, the effective area of head group and the head group changes. Israelich gave a very important equation considering this amphiphilic molecule as a cylinder. He said that there would be parameter called packing parameter P, which can be equated to V by A0L, where V can be thought about the volume of the alkyl chain and A0 can be thought about the head group area and L is the alkyl chain length. Now based on the value of P, you can predict the shape of the self-assembled structures. So suppose if you take single chain lipid with very large head group area, which is prevalent in SDS in low salt condition, then you will see that the packing parameter is less than one third, less than 0.33, and that forms a critical that gives a critical packing shape like cone, and it forms eventually a spherical micelles. But if you increase salt amount in the SDS micelle, so effectively the head group area becomes small because of the charge screening pulsive force. Then in that case critical packing parameter increases and it lies between 0.33 to 0.5, and the critical packing shape becomes truncated cone and can eventually form elongated cylindrical micelle or worm-like micelles. But in the other case, like double chain lipid case, you can have something like truncated cone based on the packing parameter value, and it can give bilayer structure, or it can have, if you have packing parameter exactly 1, then it gives a cylinder, and which means it forms planar bilayer structure. Or if the packing parameter increases to more than 1, and that is possible when you have double chain lipid but very small head group area, then you can actually form inverted micelles. Many of the common representations about micelles seen in the textbooks are wrong. You have to remember that there is no dense core and the head groups are not always perfectly arranged, and most of the normal surfactants, the micelles are not shape persistence, means they don't stay intact like always. So this more realistic is the bottom picture representation, where you can see the hydrophobic tails are actually arranged randomly, and water molecule can actually penetrate quite deep inside, and it is not that it is completely hydrophobic inside. Like if you take pluronics micelles, which is a non-ionic micelles, up to the 30% of the core is actually water. But however, in the ionic micelles do have a hydrophobic core, and you can see also a stern layer where till that point water molecule can actually penetrate, and you also have Goy-Chapman layer where you also have that comes between 10 to 100 nanometer, and in the Goy-Chapman layer you can have your cation, so cationic counterion or anionic counterion, whichever case it is applicable. This micellization from monomer to forming a micelle with some particular aggregation number. So this entire surfactant aggregation is a cooperative process, and more or less you will have discrete number of molecules coming together. So you have a discrete aggregation number, and the transition from single amphiphilic molecule to micelle is that particular onset of aggregation is known as critical micellar concentration, and this is given as delta Z micelles equals to minus RTLN CMC. And another important thing you can see, the lot of property which actually changes beyond after micellization and before micellization. Just like your osmotic pressure increases after micellization, but turbidity also increases after micellization. But there are also few things which reduces upon micellization, like conductivity reduces. Next, we look at the difference between ionic micelles and non-ionic micelles. In ionic micelles, if you see on the left hand side, the phase diagram, you see there are two lines. So one is dependence of micellar solubility on temperature, and another one is the dependence on CMC on temperature. Both increases with temperature, but the slopes are completely different. But at a particular temperature, both the curve they meet at that point, and that is called Kraft point, and below the Kraft point no micelles are possible. So but in the case of non-ionic micelle, you see it has a completely opposite temperature effect. Like if you go below Kraft temperature, there is no micelle possible, but if you go beyond a particular temperature in non-ionic micelles, they kind of all the micelles precipitate, and that's something called cloud point. So they are ionic micelles and non-ionic micelles, they completely show different behavior upon changing temperature. One thing, due to Kraft point, the transition is very sharp, but in the case of non-ionic micelles, due to cloud point, the transition is much less sharp. Another two very important differences we see when we compare ionic micelles and non-ionic micelles, they are aggregation number and CMC. We do see that the number of amphiphilic molecule which is there in a particular micelle, that's aggregation number. The aggregation number is low in ionic micelles as compared to the non-ionic micelles. Let's say if you take a CTAB on or the SDS micelles, which are ionic micelles, you would hardly get an aggregation number of something like 70, 100, 200 maximum. But if you, in the same if you take Tritonics, which is a non-ionic micelles, and you look at the aggregation number of that, that will be in thousands. So also, if you look at the CMC normal of the non-ionic micelles are actually much more lower. It will go 10 to the power minus 4, 10 to the power minus 5 molar. However, if you look at the ionic micelles, they are little higher. It will be in 10 to the power minus 2 molar or 10 to the power minus 3 molar. So it's in millimolar, and that is in sub-millimolar. So there is a difference, and that has to do with the when we have these head groups close together, so there is this repulsion happening, and that repulsion in ionic micelles, that actually causes this difference in terms of aggregation number and CMC. Next, we'll learn a very important technique that is called Langmuir isotherm, that is very useful for probing the morphological transitions between different self-assembled structures. So what we do here is that you take water, and on the top of water you make monolayer of these amphiphilic molecules by depositing the solution in chloroform and then thereby evaporating the chloroform, and you have a movable barrier so that you can actually control the entire surface area, and there is a pressure sensor which actually senses the pressure. Now, when you change this movable barrier slowly with respect to a particular time, then and you can also record the surface pressure, and if you eventually plot that in surface area versus pressure, what you will see that the surface pressure increases as you reduce the surface area, and there are some transition points, so where you can have monolayer to micelle transition or micelle to some other structures transition. So that's very important thing, and we will see why this is important in this kind of self-assembled structure. We see that you have a spherical micelles which is getting transformed into a worm-like micelles when you introduce electrolyte in the system. What happens is that electrolytes actually reduces the head group repulsion by screening. So because of that, the effective head group size becomes smaller and the P become larger. So the same spherical micelles gets into a worm-like micelle, and you can actually see that by freeze fracture electron micrograph, and you can see here the formation of fiber from this kind of worm-like micelles, and you can see the increase in viscosity as you are going for spherical to asymmetric worm-like micelles. You see there is, if you don't put electrolyte, the viscosity doesn't increase, but as you put electrolyte like potassium iodide in pyridinium based amphiphile, you see depending on the concentration of the Ki, the viscosity increases. So when we characterize these micelles, so what are the things we are looking for? So we look for the entire size of the micelles that we can do, that we can actually look at from TEM. We can do dynamic light scattering, we can do surface tension measurement. So overall, whatever you get the idea from different techniques is the overall structure, size, shape of the micelles. But suppose you want to look more interior of the micelles, what's going on in the dynamics, or what's going on inner core of the micelles, how are they, how are their shape, how are they packed, then you definitely require a non-invasive technique, and SANS, small angle neutron scattering, is the best, because you can do something called contrast matching, which we are going to see now. In small angle scattering, what we do is that we take sample and we irradiate the sample with X-ray or neutron source, and the sample scatters at a particular angle, and then we detect it, and from the scattering we plot the scattering intensity with the theta, and then we get a particular pattern. We fit it to figure it out what is the shape and size of the sample. So in this case, as you see, this polystyrene, which is a hydrophobic polymer, polystyrene, and this pyrrolidone is another polymer which is polar. So when you make a blocko polymer of that, they form inverse micelle in toluene. So that means pyrrolidone unit goes inside and polystyrene is outside. Now what we do is that if we have to measure small angle neutron scattering, it is important that we do differentiate the micelles with its surroundings, or different portion of the micelles with different neutron scattering. So that you can do by deuteriating preferentially one portion. So in this case, if you want to really look at inner structure of this blockopolymer micelle, so you can deuteriate this polystyrene part. So what you will get a differential small angle neutron scattering. So that will actually give you more idea about the inner structure of the micelles. You see that when you make this blockopolymer and with a deuteriated polystyrene unit, and when you record it in toluene or you record it in deuteriated toluene, you get completely different kind of information. For example, if when you do it in toluene, then the only deuteriated part is the polystyrene part. So basically you get the scattering coming out due to the corona of the micelle. But when you do the SANS in toluene, deuteriated toluene, then the only difference of the solvent and also the periphery is the core, which is non-deuteriated. So you get scattering due to the core, and from there you can actually find out the radius of the core and radius of the corona and also the entire micelles. So this is very important and interesting technique. Next important morphology we are interested is in bilayer and vesicle. So that is possible only when you have dual hydrophobic chain and with a polar head group, and normally this is prevalent in lipid, and that's why lipid forms this lipid bilayer, which also closes itself into liposome. And this is you see in bilayer membrane in our body. The most important thing is that these bilayer membrane are semi permeable, and this lipid molecule, they can actually move laterally, but the flip flop motion, means going from the exterior layer to the interior layer or the vice versa, is prevented. So this is called flip flop motion, and flip flop motion is not that, it's actually regular, and that actually maintains the asymmetry of the bilayer membrane. There are definitely some important bioactive group present in exterior membrane, and they maintain because of this prevention of flip-flop motion. When we look at this vesicle formation mechanism, is basically two-step process. First is that the formation of the bilayer, and the next thing is that closing of the bilayer into vesicle. So now if you have a planar bilayer, the packing parameter is equals to 1, and for the vesicle it has to be less than 1. So there is an equation given, that is the packing parameter, which is V by AL, that can equate to 1 plus HL plus KL square by 3, where H and K, they are different interfacial mean curvature or Gaussian curvature. So you see that cartoon image, it shows that again this A as a head group area and V as the volume, but however there are two radius which are important, and H and K can be expressed in terms of those radiuses and radii. That particular expression gives whether this will form vesicle or planar bilayer like structure. So in one of the unique cases, what has been done is that you polybutane and polyethylene oxide blockopolymer has been synthesized, and the ratio of the hydrophobic and hydrophilic block molecular weight, that has been changed, and depending on the ratio of the hydrophobicity and hydrophilicity in terms of the blockopolymer length, you can form different morphology like sphere, cylinder or bilayer, and that is what you can actually see from TM image. In the A, you can see they are forming very nice spherical micelles, and B, you can form a cylindrical micelles because the packing parameter increases because of introduction of the blocks, and then you can also go to vesicles depending on the ratio of the hydrophobic and hydrophilic block. These vesicles or liposomes, they have direct application for drug delivery. Since it contains bilayer and containing hydrophobic cavity and also polar cavity deep inside, it can take both water soluble material or oil soluble material in the specific cavity. Just like if you are talking about cadmium telluride quantum dot, which is hydrophobic in nature, you can see in the left image they would be sitting in the hydrophobic core, and you can see that on the corona area it is showing the high fluorescent images. But if you take fluorescein, which is a water soluble dye which can actually go inside and stay in the polar cavity. How a drug can be loaded in the liposome? In case you are using, instead of lipid polymers, then you can call it polymerasome. So in this case, you see, you make an aqueous drug solution and you put your polymer and it will form this vesicle, and then you do ultra filtration or gel filtration, and then you remove all the polymerasome from that, and then those vesicles of polymerasomes, they have encapsulated drug. But what happens is then eventually you take it to the site and you trigger a release, and it releases free drug. That can be burst release or sustained release. But what happens is that there is always the drugs which actually leaches out from the vesicle. That is something called permeation by Fickian diffusion. The release rate of that can be given in the relation of the permeation coefficient and the bilayer thickness and vesicle radius. So this is very important. So you see that if you have larger vesicles or you have the thick bilayer of that, then this permeation is, so the vesicle can keep your drug for longer time. So that is why nowadays people prefer the polymerasomes, because the polymeric bilayers are thicker as compared to the lipid bilayer. How in modern days people are using polymerasomes. Here it is a very nice example of stimuli responsive nanocapsule or polymerasome formation that employs cyclodextrin based host and ferrocin based guest molecule. Now it is known that beta cyclodextrin with a specific cavity size, they can only fit ferrocin, which ferrocin has Fe2 plus ferrous ion in between two cyclopentadiene unit. Now they form this complex. So what they have done in this particular example, they have attached polystyrene block copolymer to the cyclodextrin and polyethylene oxide to the ferrocin, and they form this inclusion complex, and as soon as that they form, then you have this hydrophobic hydrophilic part which eventually self assemble in water. You can load your drug molecule into it. Now interestingly, ferrocin can be oxidized, apply little more than 0.32 volt, then it can the Fe2 plus will be Fe3 plus, then but that is not a stable complex, then entire host gas complex breaks down and your drugs get released. So here they have shown that applying different magnitude voltage, they can actually attain burst release, sustained release over a time. And with TM they have shown very nice vesicles which actually gets released in the picture B and C, but again upon reversal of flying voltage, the vesicle forms back again. So let's summarize what we have learned in this module. So we have learned the micellar structure and what are the governing principle of that determines whether it will go, the amphiphilic molecule will go to spherical micelle or rod-like micelle, or let's say bilayer structure, vesicle-like structure, invited structure. So that equation is given by Israel H. Willi, and that equation is the packing parameter. So that is very important. So that this is something rule of thumb we have to take, and we learned this. And we have also seen, now if you have micelles, now micelles can come from different types of amphiphilic molecule. It can be non-ionic, it can be ionic, it can be you can influence their head group area with ionic strength of the medium, and then it can actually go into completely different geometry. So we have seen also morphological transition. So we know how to tweak the system and get into different kinds of assembly. So the next we have also looked into is characterizing these micelles. Other than a regular characterization of DLS, TEM, we have also looked at SANS technique, so that you can actually look into more interior parts of micelles. Then we moved on and we looked into vesicle, how a planar bilayer that can actually close and form vesicles. So we looked into these different aspect for that, different radius of curvature we have looked into. Then vesicles where you can entrap drugs, that what are the position, like if you have to put compartmentalized hydrophobic drugs or hydrophilic drugs, where in vesicle they will go, that we have learned. We also have known how to have how to use vesicles for drug delivery applications, and in that way if you are doing drug delivery application, there is always a problem of drug leaching due to this spermificant diffusion permeation. And when we looked into that, we also found nowadays people they prefer using a polymerasome more than a liposome. So we also, this liposome, polymerasomes, capsules, these are different terms. However, these are all enclosed compartmentalized system. We looked into all of them, and then we showed a couple of interesting examples where you can actually encapsulate drug, and you can, depending on the stimuli responsive trigger you choose, or the magnitude of the stimuli you choose, you can either have the drug release in a burst kinetics, or you can have in sustained kinetics, and you can also revert back and make the assembly very reversible. So I hope this module has been very useful for you to understand the uh, interactions and applications of non-covalent interaction in making nanostructures into spherical form, majorly in spherical form.